So before we move on to lipid metabolism and a few other things, proteins, and looking at metabolism in a little different way, thought I'd point out this sheet. There are two versions of this. One is color-coded, and the other is this black and white one. If you're going to print it out and you have a printer that's black and white, you probably want to use this one. If you have a color printer, the other one might be better. And certainly if you're looking at it online or using some sort of electronic device, then the color-coded one will probably help as well. But I thought we'd look at this one. So this has got 3 pages, and this goes through the whole process. We'll walk through one of them. So this is glycolysis with the malate aspartate shuttle, and then the activation in Krebs, and then the summary with an explanation. And then this would be the glycerol phosphate shuttle. And then this one would be looking at anaerobic glycolysis. And so maybe it wouldn't hurt to spend a minute or two just looking at this in general. So what I'm going to do is I'm going to look at the full screen mode, and we can see the whole thing then. So what we want to do is to look at this. This is the malate aspartate shuttle, okay, with the complete oxidation of glucose. So we start with glucose, right? 6-carbon sugar. We add 2 ATP. You can see the arrow in. Goes down. It breaks apart into a pyruvate here that's got 3 carbons, and a pyruvate here that's got 3 carbons. After it breaks, for each pyruvate, I get 1 NADH and 2 ATP. Now that 2 ATP, right, that's what I use. It's the only time I'm going to have to use ATP in this process here. For the NADH I get out, right, I get 1 each, but this is outside the mitochondria. Notice this is in the cytosol. And I have to get the NADH into the mitochondria to make ATP. The way I get NADH in, it can't diffuse in by itself because it's a large polar molecule. So I transfer it to the malate aspartate shuttle, basically. That moves the energy into the mitochondria and transfers it back to an NADH. Okay? And I do that on both sides. The pyruvate can get into the mitochondria itself. And then we activate pyruvate by turning to acetyl coenzyme A. I get 1 NADH in that process, and I get 1 CO2. Since I made 2 pyruvate molecules from my 1 glucose, right, then I have 2 acetyl coenzyme A's. Those form with oxaloacetate to make citric acid. I go through my Krebs cycle, and for every 1 turn of the Krebs cycle, I get 1 ATP, 3 NADHs, 1 FADH2, and 2 CO2s. So when I do a summary, right, we'll use different colors. So let's start here. My 2 ATP used. We'll make that a red, right? So here's my 2 ATP used. Here's my 2 ATP used, right? The NADHs that we make here, right, in glycolysis get transferred over as NADHs. So let's go ahead and just make those maybe that blue. So here's my NADH here. Here's my NADH here. So that's my NADH right here in glycolysis, right? So there's my NADHs for glycolysis. My direct ATP is right here. Here's direct. Here's direct for glycolysis. So that's this one. Okay. So I've counted for the ATP here, I believe. So how do I get these numbers? Well, for the NADH net, that's 2 times 3 is 6. 0 FADH2. So my indirect ATP is 6. My ATP total is 4 plus 6. That's 10. My ATP net is 10 minus 2. That's 8. Notice there's no CO2 at this top part, so I don't have to worry about CO2. The activation step happens in the mitochondria. So in the activation, I get 1 NADH for each pyruvate molecule. So that's this one right here. In the activation, my NADH, I'm already in the mitochondria. I don't have to worry about a shuttle. All right, we'll also account for the CO2. So here's my CO2 here, and here's my CO2 here. And so that's, again, this is the activation step. See it right here, activation. So here's my 2CO2 and my activation step. Activation's pretty easy though. I don't have any ATP used or direct. I don't have any FADH2. So my indirect ATP is how I count. 2 times 3, 2 NADHs times 3, my energy equivalents, gives me 6 ATP indirect. My total is 6 plus 0, that's 6. My net is 6 minus 0, that's still 6. And we already accounted for those 2 yellow CO2s that we drew. Now we're going to look at the Krebs cycle. So in the Krebs cycle, we haven't used pink yet, right? So I get 1 ATP in my Krebs cycle for the pink. So that's these 2 ATP, right? 1 per turn, 2 turns for glucose. So there's my 2. My NADH, we haven't used purple. So here's my 3 NADHs here, my 3 NADHs here. That gives me 6 NADHs right there. I haven't used yellow yet. My FADH2s, a little hard to see, but here's my 2 FADH2s in yellow right there. And then we'll use gray. Those are my 2 CO2s. And here's my 4 CO2s right here. So we can total that now. Right, so my ATP direct is 2. My NADH net is 6. 6 times 3 is 18. Hold that off on the side of your head. FADH2 is 2. 2 times 2 is 4. We were holding 18 here and 4 here. That's total 22 indirect. 22 plus 2 is 24. That's my total. 24 minus 0 is 24. That's my net ATP. And then my CO2 is 2 plus 2. That's 4. That's all for Krebs. Now we can add everything up. All right, ATP used is 2. ATP direct is 4 plus 2 is 6. NADH net is 2 plus 2 plus 6. That's 10. FADH2 net is 2. ATP indirect, remember there's 2 ways to do it. 6 and 6 and 22 is 34. Or 10 times 3 is 30. 2 times 2 is 4. That's 34. My total, 10 plus 6 plus 24 is 40. Or 34 plus 6 is 40. 40 minus 2 is 38. And then 8 plus 6 plus 24 is 38. And then I broke down glucose as completely as possible and liberated all the carbons. The 6 carbons came off of CO2, so I have a 6 carbon total. So that's kind of how you look at it, and you should have a pretty good idea of how this works for all of these. So if we look at the glycerol phosphate shuttle, it's the exact same thing, right? So here's the malate aspartate, except this NADH here, where that hand is circling, the 2, becomes FADH2. And what you'll notice on the drawing, so again, this is a consequence of the different shuttle and having FADH2 in the mitochondria instead of NADH. So you should understand the difference in the glycerol phosphate shuttle versus the malate aspartate shuttle. You should understand the difference between total and used, and the active versus passive transport, and where it's at. Okay, so as an example, the glycerol phosphate shuttle is the same thing as saying in skeletal muscle, which is the same thing as saying active transport mechanism, which is the same thing as saying 38 total, which is the same thing as saying 36 net. If you look at the malloaspartate shuttle, that's the same thing as saying in liver or heart muscle. It's the same thing as saying 40 total, 38 net, passive transport mechanism. So all those are refer to the same pathway. The anaerobic glycolysis is also shown. It's pretty simple. So it shows what we have here. And we see that the NADH gets used up here, even though we made it. It gets used up, so we don't even count it. So it's 0 here. Here's my 4 direct ATP. Here's my 2 ATP to start that we use. And, this one's pretty simple. Also notice all of this occurs in the cytosol, not the mitochondria, because mitochondria need oxygen. We don't have oxygen for this process. So that was just to show you this little handout that's posted, and you probably want to take a look at that and study that some. Now, there are other ways other fuel sources, I guess is the best way to say, that allow us to get ATP as well. And actually, triglycerides have more bonds, they have more free energy, and so they produce significantly more ATP. While we just looked at the aerobic metabolism of glucose, which gives us at most 40 ATP total, a triglyceride is usually 10 times that at least, and they give us 400 to 500 ATP. So when we break down a triglyceride, there's a couple things that have to happen. The first step is called lipolysis. We store our lipids as triglycerides in our adipose tissue. And so the first thing is we have to take the triglyceride and break it down. We use an enzyme called lipase, and that happens in the cytosol. And lipolysis is when we take the triglyceride and break it down into a glycerol plus 3 fatty acids. And remember, fatty acids are these long, typically even number carbon chains that are made up mostly of methyl groups with a carboxyl group at the end. Now, the glycerol we can use for other things. Theoretically, it could go and work basically half as in glycolysis, but because it can be converted to an intermediate, but we typically don't do that. And so most of the energy we get from a triglyceride, if not all of it in a typical day, would be fatty acids. So once we have those fatty acids, we have to use those fatty acids for energy. And the way we do that is we break the fatty acids into 2 carbon units. So just to kind of show you, I'll just write them here. So let's say we had, and this is obviously a lot shorter than most, a fatty acid that had 6 carbons long. So here's my 6 carbon fatty acid. Each carbon is represented by a C. And when you look at this molecule, then this first bond right here, this is called the alpha bond. Right? And alpha is like a little fish. I didn't draw it very well there. It looks like this without a little closed tail. And then the second bond is called the beta bond. And the beta bond is like a handwriting B. And what we do is we chop the 2 carbon units off at the beta bond. And then we take these 2 carbons and it becomes acetyl coenzyme A. So it's got the acyl unit, which is just 2 carbon, and we put a coenzyme A on it, and it becomes acetyl coenzyme A. So there's a couple ways to make acetyl coenzyme A. We saw before with glucose from pyruvate, you can make acetyl coenzyme A. When pyruvate moves to the mitochondria, it becomes activated. Or you could do lipolysis to a triglyceride, take the fatty acid, and chop off that 2 carbon unit, and then convert that to acetyl coenzyme A, and then use that in the citric acid cycle. Now, once this 2 carbon unit is gone, so I'm just going to erase it because we made it acetyl coenzyme A. Now I have 4 carbons left, and then I have a new alpha, a new beta bond, and I chop that off there, and now this 2 carbons goes and becomes acetyl coenzyme A, and this 2 carbon becomes acetyl coenzyme A. Well, our example was just a 6 carbon molecule. In real life, most of our fatty acids are 16, 18, 20, 24, 26, all the way up to 32 carbons long that we can use to break down for fuel. So we can get a lot of energy from one fatty acid, and then we have 2 more in that triglyceride. Once it becomes acetyl coenzyme A and it goes to Krebs, Krebs is Krebs. It doesn't care whether the acetyl coenzyme A came from pyruvate or from triglyceride through beta oxidation. All it knows is, okay, I got an acetyl coenzyme A, I'll go through it. So that would give me that 1 acetyl coenzyme A in 1 turn of the Krebs cycle would be the same yield we had before. You get 2 CO2s, 1 direct ATP, 1 FADH2, and then 3 NADHs, right? So for every 2 carbon unit you chop off in beta oxidation, you get 12 ATP total, and then 2 CO2. So now it should make sense why fatty acids come as 2 carbon units in even numbers. If we had a 17 carbon chain fatty acid, what would you do with that extra carbon? There are a couple in the body, but the vast majority of them are even numbered carbons. And again, once you get to the citric acid cycle, the NADH and the FADH2, right, then go into electron transport to make ATP. That's all fine and good. There's one problem with lipid metabolism compared to carbohydrate metabolism. Remember, carbohydrate metabolism did have that one advantage of being able to do anaerobic glycolysis. All of the big energy we make, beta oxidation and citric acid cycle, occur in the mitochondria. So generally speaking, you need oxygen available in the mitochondria to break down fat, and that is an issue. Although we're never truly aerobic or anaerobic in our lives, except I guess you're truly anaerobic when you're dead. But outside of that, we're always have some aerobic capacity. One quick note: this has led some people to mistakenly say, well, if you want to burn fat, you need to exercise at a low level. That's not true. Yes, you burn a greater percentage of fat at lower levels of exercise, but the total amount of fat you burn is higher at pretty much every level of exercise intensity. So the higher the intensity you go up, yeah, you might only burn 20% fat, but you're burning 3 times as many calories, so you're burning more fat than you would normally. And it's probably not even a question of how much fat versus carbohydrate you burn anyway. It's probably more a question of the total caloric burn versus the total caloric intake in terms of body composition, especially. So that's how we use triglycerides to make energy. As mentioned before, we can also use proteins for energy. Now, proteins have a couple issues, and we don't typically use proteins for energy, and there's three big reasons why, probably. Number one, we like to save proteins for other things. Carbohydrates and lipids are our primary fuel source, and there's not a whole lot of other things they do. Yeah, there's a few other structural things and signaling things, and yeah, steroids, which is a different type of lipid anyway, are used for hormones and signaling molecules, and the phospholipids make up the membrane, so they're important, and things like that. But for the most part, there's not a whole lot of functional diversity in terms of lipids or carbohydrates. So we try to save those and spare those for energy. Proteins are used for pretty much everything else. And we can't use a carbohydrate to make an enzyme typically. And we can't use a lipid to make a muscle. And so we like to spare our protein when we can so that we have enough to do the other things protein needs to do. The other problems with protein is that they're not very efficient, especially at the beginning. So people are pretty efficient at breaking down lipids, pretty efficient at breaking down carbohydrates, not very efficient at breaking down proteins. And so, from a weight loss standpoint, that's why high protein diets tend to work, especially for about a half a year or so, because people aren't very efficient at breaking them down. So if you were to eat 1,000 calories of lipid, you need to burn probably over 900 of them at least, because it takes some energy to process it, but not that much. And the same thing is true for carbohydrate. But if you were to eat 1,000 calories of protein, it would be much more difficult to break down, and you would only have to burn 600 calories of that protein because you'd spend 400 calories breaking down protein. And as you eat more protein and get used to those high protein diets, then your body will become more efficient, and which if you're trying to lose weight, it's not a good thing. So for protein catabolism, breaking down proteins and using them for energy, we can't directly use proteins for energy, so we have to do a couple things to them first. The first step to breaking a protein down is proteolysis, protein, lysis, break apart. And basically we need to break the protein into monomers. The enzymes are called proteases. And what we do is we take the polypeptide, this protein, and break it down into amino acids. Now, certain amino acids are ketogenic and certain amino acids are glucogenic. And we'll talk a little more about that in a minute. But in order to use amino acids and convert them into material that we can use for energy, we have to deaminate them. And deamination is basically taking the amino group and removing it so that we're left with this organic acid. Now, this organic acid can be made into glucose or into acetyl coenzyme A, basically. But what we're left is ammonia, right? So we've removed the amino group, put another hydrogen on it, we've got ammonia. That's not good. And ammonia is toxic to the body. So basically what we do is we convert it to ammonium ion and then urea, and then, usually not excrete at all, but excrete some of it. So the deamination is part of the higher energy requirement of breaking down protein. Also, it's one of the more dangerous things you can do in terms of metabolically, because it puts an overload on your kidneys to process that. So once we have those amino acids, now that we've removed the amino group from it, we can now make energy from it. Some of them, like I said, are called glucogenic, and we can literally convert them to glucose. Once they're glucose, right? And the body doesn't differentiate between a glucose molecule you have from a soda and a glucose molecule you made from amino acid. And so everything that we know about glycolysis and the catabolism of glucose fits Or you could have ketogenic amino acids, where basically you make acetyl coenzyme A. And then you could go in the Krebs cycle like we saw for the lipids. One of the nice things about protein is this as well. I mentioned in a previous video that humans can make excess carbohydrate into lipid, but we can't reverse because we don't have the enzymes. So we can't make lipids into carbohydrate, even if it was originally a carbohydrate, right? We can't go backwards. So if we run out of carbohydrate fuel, and we really can't store that much carbohydrate relative to lipid, we need to find carbohydrates some other place, because again, our nervous system especially has a need for carbohydrate in their metabolism. And so if you don't have it in your diet and you don't have carbohydrates stored, you can't make it from fat, so the only place you can get it is from protein. And if you're on a low caloric diet and your body's not getting sufficient glucose, it will start to break down protein and turn it into glucose. And you might lose a lot of weight, but eat your muscle literally to provide the metabolic substrate for your nervous system is probably not the best way to go in that regard. So if you eat enough protein in your diet, you'll do that first. But you do have to be careful in starvation situations. And the difference between starvation and fasting, generally speaking, is fasting is voluntary, starvation is not. But in either case, whether you're purposely not eating things or just can't eat things for various reasons, you're gonna potentially go into this protein metabolism where you're eating your protein muscle up, because that's the most available source of protein in your body, and to make glucose out of it, but it's very difficult to replace that muscle that you've destroyed. So something to think about. But we can make proteins into energy as well, but we don't do it very often. Depending on what source you read, most scientists will say at least 95% of your energy expenditure in a day comes from non-protein sources, although that changes a little bit now as people are on ketogenic or high protein diets where they're purposefully trying to get their body to use protein for fuel. So this kind of summarizes everything we talked about and how we aerobically can break down these things for energy. I'm not going to review them. You can look at them. The other way to look at this is, what are the sources of energy for the body? Okay, and so the first source of energy is ATP. So ATP is the most immediate source of energy for your body. Okay, and if you stored all the ATP you used in a day, you probably weigh another 350 to 400 pounds. That's how much ATP we would use over a day if we stored all of that ATP at once. We don't, and there's a number of other reasons why, but one would be the efficiency. You just wouldn't function as well if you were carrying an extra 300 or 400 pounds of ATP. So what we do is we store our energy as other molecules that we can fairly easily and quickly convert to ATP when we need it. So the number one source of energy, the most immediate source of energy, is ATP. But we only have enough ATP to do about, 5, 6, no more than 10 seconds of exercise. The second source of ATP is creatine phosphate. And creatine phosphate is a useful molecule where what we do is, I'll show you. So let's say that this is the mitochondrial membrane, all right? And, this is one of the big problems that most books don't address. So this blue line that's not drawn very straight is the mitochondrial membrane. Most of the ATP we make, as we've seen previously, is made in the mitochondria. But to be honest, most of the ATP we use is used outside the mitochondria. And we have this big problem. We have ATP in the mitochondria that cannot pass the mitochondrial membrane to get out. And so how do we get that high energy out of the mitochondria into the other places in the cell where we need it? Well, what we have is we have this system where we have a molecule that's called creatine. So I'm just going to put CR, that's creatine. Okay. Some of you might recognize creatine as a supplement that people take for, bodybuilding and other activities. And what happens is, this creatine, what we do is we say, okay, we're going to take a molecule of ATP. So creatine plus ATP. And what happens is ATP says, you know what, I'm going to give you my phosphate. So what it does, it's going to give creatine one of its phosphates. And when it gives creatine one of its phosphates, that now becomes CRP, creatine phosphate. So that's what we see down here, creatine phosphate. Plus now we have ADP. So that ADP that we have here, that can go to the electron transport chain and make ATP, because all we have to do is now put a phosphate on it. And this creatine phosphate molecule can actually diffuse out of the mitochondria and go into the cytosol. And what happens in the cytosol is we just reverse this. So creatine phosphate finds an ADP molecule outside, and that converts it to ATP, and then creatine goes back in to the mitochondria and just keeps on doing that. This is called the creatine phosphate shuttle, and it's the way that we get ATP energy out of the mitochondria across the mitochondrial membrane without having to move the ATP directly. So the mitochondria basically has its own ATP store. It doesn't leave the mitochondria. And the cytosol has its own ADP and ATP store that doesn't leave the cytosol. And so creatine phosphate is also an energy buffer. And what it does is, as ATP starts to be used up, we can transfer the creatine phosphate to ATP. And that adds another 10 seconds, right? So you got a couple seconds of ATP energy, and then you got a couple more seconds of creatine phosphate energy, and that's not enough. And so where do we get our next bit of energy? That comes from glycolysis. Remember, glycolysis can only happen with glucose. Right? We actually can convert some sugars to glucose, like fructose. We don't convert to glucose, but mostly the other sugars we do, 6-carbon ones at least, and use our simple sugars to do the anaerobic glycolysis. And that gives us another 30 seconds worth of exercise. And people that are doing, high level anaerobic work, right, maybe can do it for about not even 30 seconds. Okay. So that's the next level. So we go ATP is 1, creatine phosphate is 2, anaerobic glycolysis with glucose, not a very good 3, but there it is 3. And 4 is going to be our long-term complex carbohydrates and our stored lipids, right? So our stored carbohydrates and our stored lipids. How much energy do we actually have? The average person maybe can store about 400 grams of carbohydrates in their body. So that's not very much. It's like a pound. The stored lipids, though, most college-age males are between 10 and 20% body fat, and college-age females are probably between 15 and 25% body fat. So someone who weighed 100 pounds and had 20% body fat would have 20 pounds of lipids. Now, that all wouldn't be stored lipids, but much of it would be. And so our long-term energy storage, our aerobic energy storage, literally can provide energy for, indefinitely and at levels of exercise for long periods of time. I don't know if they still do it, but they used to run a race in Colorado where literally people just ran circles around a track for 24 hours. And they just counted how many laps they could make in 24 hours. And people just ran for 24 hours straight, basically. So you can exercise for really long periods of time if you need to. And obviously you have to be properly trained and prepared and, have an intake of food and things like that. But that's kind of the way our energy stores work. So this is the summary of what I talked about. So ATP, inefficient storage, right? Just for one muscle contraction, you use 10 million ATP molecules, right? So again, you got about 350 pounds per day. So we regenerate our pools. Then we talk about creatine phosphate, and the phosphocreatine shuttle, right? So we go from a couple seconds to a couple more seconds. So together, it's maybe 10 or 15 seconds. The simple sugars, right, that we talked about for anaerobic glycolysis, and that would be our blood sugar. Our complex carbohydrates stored as glycogen, right? About 4,000 grams in terms of that. So you probably have a couple hours of moderate exercise. And then lipids are your most calorically dense energy storage. It has less weight because of hydration, really lack of hydration. Lipids are said to be anhydrous, meaning they don't store water. The carbons are more reduced, and the average person has at least 10,000 calories of fat, right? And that's more than enough to run 100 miles, or if you're a bear, sleep for months in hibernation, right? So, wrong way. So that completes the metabolism notes. Pay attention to how we use especially glucose for energy, but you're responsible for all of this. And, take a look at, especially the handout. Probably wouldn't be bad to practice writing the handout and, looking at the different pathways. So that's it for the metabolism lectures.